Welcome to GE Fun Facts Online. For the better part of a century, humanity has been locked in a microscopic war. Our primary weapon against bacterial invaders has been antibiotics, miracle drugs that have saved countless lives. But we've used them so relentlessly in our medicine, in our agriculture, that our enemies are adapting. We are now facing the dawn of a post-antibiotic era, a terrifying prospect where common infections could once again become death sentences. But what if our entire strategy has been wrong? What if, instead of waging a war of annihilation, we could simply disarm the bacterial armies, rendering them harmless without a single casualty? This isn't science fiction. It's a revolutionary new frontier in medicine called quorum sensing disruption, and it involves hacking the secret social network of bacteria. So, let's dive right in. What exactly is this bacterial social network? The scientific term is quorum sensing. The word quorum itself refers to the minimum number of members needed for a group to conduct business. It's the same principle for bacteria. They use a sophisticated system of cell-to-cell communication to take a head count of their own population. Why? Because launching an attack on a host organism, like you, is a numbers game. A few dozen bacteria trying to cause an infection will be swatted away by the immune system in an instant. It's a suicide mission. A smart pathogen waits. It multiplies quietly, staying under the radar, until its population reaches a critical mass, a quorum. Once the head count is high enough, they launch a coordinated, synchronized attack, unleashing a barrage of virulence factors all at once. It's a strategy of overwhelming force, and it's orchestrated entirely by quorum sensing. The language bacteria use for this is purely chemical. Individual bacteria are constantly manufacturing and releasing small signaling molecules into their environment. These are called autoinducers. You can think of them as constant, low-level chatter. At low population densities, these molecules simply diffuse away. The chatter is too faint to be meaningful. But as the bacteria divide and the colony becomes more crowded, the concentration of these autoinducer molecules skyrockets. Eventually, it crosses a critical threshold. At this point, the molecules begin binding to specific receptor proteins either on the surface of or inside the bacterial cells. This binding event is the trigger. It initiates a complex chain reaction, a signaling cascade that switches on a whole battery of genes. These are the genes responsible for the bacteria's most dangerous behaviors, the ones that are only effective when undertaken by a large, synchronized group. The list of behaviors controlled by quorum sensing is a greatest hits of bacterial pathogenicity. First and foremost is the production of virulence factors. This includes a vast arsenal of weapons. Toxins that poison and kill host cells, enzymes like proteases and elastases that degrade our tissues and break down our defenses, and systems that help the bacteria steal essential nutrients, like iron, from our bodies. The opportunistic pathogen Pseudomonas aeruginosa, a major cause of hospital-acquired infections and a severe threat to cystic fibrosis patients, is a master of this. Its quorum sensing network controls the production of dozens of virulence factors, including a toxin called pyocyanin, which generates oxidative stress and causes widespread tissue damage. Another critical behavior is biofilm formation. If you've ever felt that slimy film on your teeth in the morning, you've met a biofilm. It's a structured community of bacteria, encased in a fortress of their own making, a gooey matrix of sugars, proteins, and DNA. This slimy shield is a marvel of microbial engineering. It protects the bacteria within from antibiotics, sometimes making them up to 1,000 times more resistant. It also physically blocks immune cells from reaching them. Quorum sensing is the master conductor for biofilm construction, from the initial attachment to a surface to the maturation of the complex three-dimensional structure. This is why infections on medical devices, like catheters, artificial joints, and heart valves, are so notoriously difficult to treat. They are almost always biofilm infections. But it doesn't stop there. Quorum sensing also governs motility and swarming, allowing bacteria to spread rapidly across surfaces and colonize new territories within a host. It can control bioluminescence, the production of light, which is how this entire field was discovered in the glowing bacterium Vibrio fischeri. It even regulates processes like the transfer of genetic material between bacteria, which is one way that antibiotic resistance genes can spread so quickly through a population. Now, just as humans speak different languages, bacteria have evolved different dialects for their chemical communication. The systems they use often depend on their basic physical structure. Gram-negative bacteria, which have a thin cell wall sandwiched between two membranes, primarily speak a language based on molecules called N-acyl homoserine lactones, or AHLs. The classic model for this is the Luxi/Luxar system, first studied in that glowing Vibrio fischeri. A synthase enzyme, Luxi, produces a specific AHL molecule. These molecules are small and lipid-soluble, so they can pass freely across the bacterial membranes. As the population grows, the AHL concentration builds up both inside and outside the cells. Once it hits the threshold, the AHL binds to its partner, a receptor protein in the cytoplasm called LuxR. This AHL receptor complex then acts as a transcriptional activator, binding directly to the DNA and switching on target genes. The beauty of this system is its specificity. By changing the length and modifications of the acyl side chain on the AHL molecule, different species create unique signals, ensuring that they are mostly talking to their own kind. In contrast, gram-positive bacteria, which have a much thicker exposed cell wall, typically use a different class of signals, small, modified peptides called auto-inducing peptides, or AIPs. A well-studied example is the AGR system in Staphylococcus aureus, the bacterium responsible for everything from skin boils to life-threatening sepsis. In this system, a precursor peptide is synthesized, modified, and actively transported out of the cell. The AIP then accumulates in the environment. Once it reaches a high enough concentration, it's detected by a two-component sensor system embedded in the cell's outer membrane. This detection triggers a phosphorylation cascade, a chain reaction where phosphate groups are passed from one protein to another, like a series of falling dominoes, which ultimately activates a response regulator protein inside the cell. This protein then turns on the expression of virulence genes. The diversity in the amino acid sequence of these peptides allows for an incredible degree of specificity, sometimes even distinguishing between different strains of the same species. But perhaps most intriguing is the discovery of a universal language, a bacterial Esperanto that allows for interspecies communication. This is mediated by a molecule known as AUTOINDUCER2, or AI2. The enzyme that makes AI2, called Lux, is found in hundreds of different bacterial species, both gram-negative and gram-positive. This has led scientists to believe that AI2 acts as a general signal for bacterial density, allowing diverse species within a mixed community, like in the human gut, on the skin, or in a complex dental plaque biofilm, to get a sense of the total microbial load and coordinate their behaviors accordingly. This intricate understanding of bacterial communication has opened up a thrilling, new therapeutic avenue. If bacteria need to talk to be dangerous, what if we could just hang up the phone? This strategy is known as quorum quenching. Instead of the brute force approach of killing bacteria, which drives the evolution of resistance, we can simply silence them. Scientists are pursuing three main strategies to achieve this. The first is to sabotage signal production. This is the most direct approach. Stop the conversation before it even begins. Researchers are hunting for small molecule inhibitors that can bind to and deactivate the synthase enzymes that produce autoinducers, like the Luxi-type enzymes in gram-negative bacteria. If the bacteria can't make the signal molecules, their entire pathogenic program fails to launch. The second strategy is signal degradation. If the signals are already being produced, we can intercept and destroy them in transit. Nature has already provided us with the tools for this. Scientists have discovered a variety of quorum quenching enzymes produced by other microbes that can do just that. For the AHL signals, there are two main types. AHL lactanases, like the AIIA enzyme from Bacillus species, work by hydrolyzing the ester bond in the homoserine lactone ring of the molecule, effectively breaking its structure and inactivating it. Then there are AHL acylases, which cleave the amide bond connecting the side chain to the ring. Both methods neutralize the signal. The therapeutic potential is obvious. Imagine a cream containing these enzymes that you could apply to a wound to continuously mop up bacterial signals and prevent an infection from taking hold. The third, and perhaps most sophisticated strategy, is to block signal reception. Here the signals are produced and they fill the environment, but the bacteria are rendered deaf to them. This is often achieved using receptor antagonists. These are synthetic molecules, designed in a lab, that are structurally similar to the natural autoinducers. They are close enough in shape to bind to the receptor proteins, but different enough that they fail to activate them. They act as competitive inhibitors, essentially sitting in the receptor's active site and blocking the real signal from binding. It's like putting a dud key into a lock. It fits, but it won't turn, and it prevents the real key from being used. A fantastic example is a synthetic molecule called metabromothiolactone, or MBTL. It has been shown to powerfully inhibit a key quorum sensing receptor in Pseudomonas aeruginosa, leading to a dramatic reduction in virulence factor production and biofilm formation, both in the lab and in animal models of infection. So why is this approach generating so much excitement? The primary advantage is that quorum quenching is an anti-virulence therapy, not an antibiotic one. It doesn't threaten the bacteria's survival. It only neutralizes their weapons. This is crucial because it is thought to exert far less selective pressure for the evolution of resistance. A traditional antibiotic creates a life-or-death situation. Any bacterium with a random mutation that allows it to survive will rapidly multiply, and soon you have a fully resistant population. But a quorum-quenching agent doesn't kill. The bacteria can still live and multiply. The evolutionary drive to develop a mutation to overcome the signal jammer is theoretically much lower. Moreover, quorum quenching offers a powerful way to combat our existing antibiotic resistance problem. It can act as a booster for our failing antibiotics. Many bacterial resistance mechanisms, like the formation of those protective biofilms or the action of efflux pumps that actively spit antibiotics out of the cell, are themselves regulated by quorum sensing. By inhibiting quorum sensing, we can dismantle these defenses. We can break up biofilms, exposing the now vulnerable bacteria within. We can shut down the efflux pumps. Studies have shown that combining a quorum sensing inhibitor with a traditional antibiotic can have a synergistic effect, restoring the antibiotic's effectiveness against strains that were previously considered resistant. And the applications extend far beyond human medicine. In agriculture, bacterial pathogens cause billions of dollars in crop damage annually, and they use quorum sensing to do it. Quorum quenching offers a novel, environmentally friendly alternative to chemical pesticides. Scientists have engineered plants, like potatoes and tomatoes, to produce AHL-degrading enzymes, making them inherently resistant to certain bacterial blights. Another approach is to use beneficial, non-pathogenic bacteria that naturally produce quorum-quenching compounds as biocontrol agents, spraying them on crops to protect them from disease. In the industrial world, biofouling, that accumulation of microbial slime, is a costly menace. It clogs pipes in water treatment facilities, reduces the fuel efficiency of ship hulls, and contaminates food processing surfaces. Quorum quenching agents could be incorporated into paints, coatings, and filtration membranes to prevent biofilms from ever taking hold, saving industries billions of dollars and reducing the need for harsh chemical cleaning agents. Of course, the road ahead is not without its challenges. The idea that quorum quenching is resistance-proof is likely too optimistic. Bacteria are the planet's ultimate survivalists. While the selective pressure may be lower, there is evidence that resistance can and will emerge. For example, bacteria could develop mutations in their receptor proteins so that the inhibitor molecule no longer binds, or they could evolve enzymes to break down the inhibitor itself. Another major challenge is specificity. Our bodies are home to trillions of beneficial bacteria, our microbiome, that are essential for our health, and many of them also use quorum sensing. A broad-spectrum quorum-quenching drug could have unintended and potentially harmful consequences by disrupting the communication within our healthy microbial communities. The holy grail is to develop inhibitors that are highly specific for the pathogenic species we want to target, without causing collateral damage. And finally, there is the long road from the lab to the clinic. Despite a mountain of promising preclinical data, the translation of these discoveries into approved human therapies has been slow. We need to ensure these compounds are stable, can be delivered effectively to the site of infection, and are not toxic to human cells. However, strategies like drug repurposing are helping to speed things up. Screening existing, FDA-approved drugs for unexpected quorum-quenching activity is a clever shortcut, as their safety profiles are already well understood. The anthelmintic drug niclosamide, for instance, was recently identified as a potent quorum-sensing inhibitor against P. aeruginosa and is now being investigated for this new purpose. In conclusion, quorum-sensing disruption marks a fundamental paradigm shift in our war against bacterial disease. We are moving beyond the 20th century mindset of indiscriminate killing and entering a new era of intelligent, targeted disarmament. By learning the secret languages of our microscopic foes, we have found a way to intercept their battle plans, to sow confusion in their ranks, and to turn pathogenic armies back into disorganized mobs. The potential rewards are immense: a solution to chronic infections, a powerful new tool against antibiotic resistance, and innovative new technologies for agriculture and industry. The silent war is far from over, but with quorum quenching, we have a new, smarter way to fight back.